So let's talk about some mediators. So as far as mast cell mediators, we have to go and think about the granule. So obviously, being a granulocyte, mast cells degranulate. They have granules that they can release, and these granules have components. So the mast cell granule matrix is formed from proteoglycans, which are these long proteins that have glycosaminoglycan, or GAG, side chains. So what we're really focused on in human mast cells is heparin. Now, heparin is the main proteoglycan we're going to find in human mast cells, and heparin helps to stabilize the tryptase enzymes, and we're going to talk about how that actually works chemically in just one slide. But inside the granule, we have so many things. We have proteases, acid hydrolysases, histamine molecules, just a big one, that are all attached to heparin that via ionic linkages to the sulfate groups on these GAGs. And now the sulfate groups are going to provide an acidic environment. But what happens when we actually degranulate? So when degranulation occurs, that acidic environment is actually going to change to a neutral environment. And in that neutral environment, the pH is going to increase, and therefore the mediators are going to be activated due to the pH change. So that's how the activators, or the mediators, are not like destroying the mast cell from inside. They're only activated once they actually leave the granule. And it's interesting to think, I just wanted to bring this up, that granulation is actually not suicidal at all. So it's totally possible for a mast cell to degranulate and then sort of restore itself over time. So just going over some of the main mast cell mediators, I want to put this image here in case you want to refer back to it, but this is a level of detail that is a little overwhelming, so I'm going to sort of bring it down a little bit and talk about the main ones. So let's actually start in the middle here, because we talked about tryptase already. So tryptase is obviously, as we know, going to be pretty pro-inflammatory, and we see it indeed recruits neutrophils, potentiates histamine release, increases cellular adhesion, and then we also have chymase. Now, chymase, as I said last time, less is known, but it does things like mucus secretion. T-cell adhesion are some of the main things it does. It's also somewhat pro-inflammatory here in the conversion of angiotensin 1 to angiotensin 2. But really, the big one that we think about with mediators in the mast cell is histamine. Now, I'll have a whole lecture on this because there's so much to talk about with histamine. But histamine's a bronchoconstrictor. It does tissue edema, vascular permeability, mucus secretion. So a lot of the main symptoms we know are going to be coming from that histamine. So these mediators that we just talked about are the ones that have been pre-stored inside. But what about the mast cell mediators that are actively synthesized? So these ones are sort of made on demand or ready to order, if you want to think about it that way. And in this case, I'm going to go over the prostaglandins and the leukotrienes. So BGD2 is this sort of big mast cell prostaglandin. It results in a lot of the same things that histamine does, so bronchoconstriction, edema, mucus, chemotaxis, all of those things. And the leukotriene CF4 and D4 are, again, going to result in very similar things. But here I've also outlined some functions that help to differentiate between these two. So these ones also enhance airway smooth muscle proliferation, which is a big part of asthma and airway remodeling. It increases secretion of IL-4 from eosinophils, increases secretion of IL-5 from mast cells. Both of these are very sort of pro-allergic inflammation. Now, mast cells can be activated in tons and tons of ways. If you look at this image over here, every single one of these is a way that a receptor on a mast cell can sort of be stimulated by an external signal. But I really want to focus on this one pathway, which is IgE dependent. So IgE dependent revolves around the Fc epsilon Ri receptor. That's sort of the main IgE receptor. But as you can see, there's so many other ways that are completely just free of of IgE as a molecule at all. But really, in the context of allergic diseases, it would be nice to focus on this one. So let's talk about IgE-dependent activation. Well, the receptor is a tetramer. It has an alpha chain which binds IgE and a beta chain which is used for signaling. So when it's bound to IgE, you can almost think that it's sort of like loaded onto, or the IgE is loaded onto the receptor and ready to interact with the antigen, as you can see in this image over here. And when it binds the antigen, that's when you get the degranulation cascade, and everything happens downstream. So this is just sort of a bird's eye view of what's going to happen, and this is something that we'll talk about way more in the lecture on the IgE molecule and the IgE receptor. But I just want you to see this from a bird's eye view. Yeah. So let's talk about some clinical applications. Well, allergic sensitization is obviously going to be one of the main ways that we think about mast cells. So mast cells help to skew that Th2 bias that we have been talking about, and it does that by releasing IL-4 near undifferentiated T cells, and that's going to polarize those T cells towards the Th2 bias. They also are able to present antigens to some T cells. They are able to sort of modify the cytokine production of dendritic cells to further skew the Th2 bias, and they also help recruit dendritic cells towards the sites of allergic inflammation. So let's talk a little bit about mast cells and anaphylaxis. Now, mast cells have alpha tryptase and beta tryptase. Now, alpha tryptase is released all the time, so constitutively, but beta tryptase is stored in the granules, and the only possible way that you can observe beta tryptase in the human body, to my knowledge, is a mast cell has to degranulate in an IgE-dependent manner. So this is why beta tryptase becomes a really strong and valuable marker in the human body, because it almost definitively tells us that mast cells have been activated by IgE, which is relevant for many of the pathologies that we'll get into later. So a common question is, why not use histamine as a marker? Now, most people think of histamine as the sort of prototypical, or I should say archetypical, allergic mediator. We take antihistamines, we don't take anti-tryptase medications. Well, the issue with histamine is that histamine is also secreted from basophils, so it's just not as specific as beta tryptase. Furthermore, histamine is degraded pretty rapidly. It only peaks in the blood after a few minutes, whereas tryptase, it lasts longer, and it's also much larger, so it's easier to sort of identify. And finally, what I want to talk about is just how we handle mast cell activation. So this is sort of a teaser for the later lecture on mastocytosis, where we're going to talk about mast cells as their own sort of distinct pathology. But there are some mast cell inhibitors that are, developing, but it's not yet really a super highly effective therapy yet. So cromolyn sodium, this is something that has been shown to weakly inhibit IgE-dependent secretion from mast cells in human lungs. But really, we've only been able to see lower histamine releases by around 10 to 20%, which is notable, but not really the level where this is going to be something you're seeing everywhere. We also see that there are KIT and stem cell factor targeting drugs, such as imatinib. So these actually have been sort of produced, but they have many side effects with somewhat limited clinical efficacy. So it's interesting to think, things might seem pretty straightforward on paper. Oh, let's just, cut off the mast cell survival factor, and that will help shut down the mast cell activity. But clinically, it's a little bit harder to get such a clean-cut function. And if you'll remember from the last lecture, we talked about lipoxins and resolvins as anti-inflammatory lipid mediators. Well, indeed, these are some things that we've seen be inhibited as a potential supplement, or administered as a potential supplement, because they sort of inhibit mast cells from degranulating through pathways that are dependent on that high affinity receptor. This may be useful for asthmatics, because we know that they are somewhat deficient in these anti-inflammatory medications as well. And finally, we have, this is more of a hypothetical treatment, but we know that mast cell receptors have these inhibition motifs. These inhibition motifs are potential targets, because if we can target it, we can sort of artificially inhibit mast cells through a way that already, or a pathway that already exists, but we would just administer an inhibitor for that function. So that is all for this lecture. And from here, I would recommend that you either watch the lecture on IgE and its receptors, or honestly, the lecture on mastocytosis is a pretty good follow-up here as well.